Hello, I am Benjamin Frey, a NOAA EPP LMR Master's Fellow at the University of Maryland Center for Environmental Science, Chesapeake Biological Lab. In this presentation today, entitled Age Validation Using Periodicity of Annulus Formation and Hardpart Microconstituents in Black Sea Bass, I am going to be presenting some of my thesis work on age validation through chemical analysis of fish hardparts. First, here is some background on age estimation, validation, and their importance. Age validation establishes the accuracy of an age estimation method. Most assessments are age structured, yet age validation remains unfulfilled for many important stocks. For example, recently an age structured assessment for Atlantic monkfish was suspended owing to the bias in age estimation, which has now motivated research on validation methods. Direct aging is one of the most common aging approaches, using the interpretation of annuli on hardparts such as otoliths, vertebrae, opercula, fin rays, or scales. On the right you can see the same black sea bass otolith sectioned on the top and whole on the bottom. Hardparts show patterns of seasonal zonation, which can be optically distinguished through microscopy as alternating light and dark zones. These zones vary in their organic concentrations. If you look in the top right, you can see the lighter translucent zone followed by the opaque dark zone, and they alternate all the way to the edge. Opaque zones typically have higher protein content and are relatively narrow in comparison to the translucent zone. They are interpreted as representing periods of lower seasonal growth, such as temperate winters, while the translucent zones represent those fast growth warm seasons. These two zones together define a ring, also known as an annulus, representing 1 year of growth. In order to validate the interpretations of annuli on hardparts, the use of known age individuals is usually required, either through lab rearing or wild tag studies. You can also apply marginal increment analysis, which suggests that if a growth increment is formed on a yearly cycle, the average state of completion of the outermost increment should display a yearly sinusoidal cycle when plotted against the season. Although there are some problems with these methods, for direct aging, inconsistent or biased interpretation of annuli, particularly in the presence of non-concentric growth and the presence of false annuli, can cause problems when reading. As for the standard validation methods, known age material and seasonal samples to support marginal increment analysis may not be available. On the bottom you can see the same Atlantic monkfish otolith, and its unusual morphology and formation of false annuli cause standard validation methods to not be viable. This brings me to our novel approach to age validation. We tested for the periodicity in otolith calcium, copper, phosphorus, strontium, magnesium, manganese, and zinc, and the synchrony of periodicity with annulus zonation, i.e., we validated aging via the seasonal absorption of these elements and compared them to the seasonal formation of annuli. Our subject species is black sea bass, which has already been validated through marginal increment analysis, and it's serving as a model species for this method to support its use for age validation of difficult-to-age fish, such as monkfish. We are testing a new validation procedure that only a few others have tried. On the bottom left, you can see a periodogram from Siski et al. 2016, which shows that 31 Atlantic bluefin tuna otolith transects had significant yearly cycles in strontium. Similarly, on the right you can see Brophy et al. 2019, which shows significant yearly cycles in phosphorus. This is for hake. Both of these models employ the Loms Gargle periodogram analysis, which is used for detecting and characterizing periodic signals in unevenly sampled data. This type of periodogram is not sensitive to global time shifts in the data, the importance of which I'll explain in the next slide, but first a short example displayed here shows on the left a standardized barium concentrations measured across the transect of an otolith, which was collected via LAIC PMS and plotted against the known age of the fish. Then on the right, the data is run through the Loms Gargle analysis to reveal any significant periodicity. The data is normalized by the residuals of the data's least squares fit around a constant reference model, and the dotted line seen on the right represents the threshold of significance. This periodogram here shows a significant period at 1.3 years. The importance of this analysis not being sensitive to global time shifts is that annuli spacing becomes narrower as fish growth tapers through their lifetime, meaning the area chemically analyzed gets smaller as they get older. This lack of sensitivity to time shifts means the Loms Gargle periodogram can be applied to our non-uniform time series. The image on the left is everyone's favorite von Bertalanffy asymptotic growth curve, and you can see how it's reflected on the right annuli formation on that otolith tapering toward the edge. To get the elemental concentrations across the transects of our otoliths, laser ablation inductively coupled plasma mass spectrometry was applied. The LAICPMS analysis provided line transects on our otoliths magnesium, calcium, strontium, phosphorus, manganese, cobalt, copper, zinc, and barium concentrations. With the LAICPMS, we were also able to get higher resolution elemental 2D maps to help reveal annual patterns on section otoliths. Here we have some 2D elemental maps of a black sea bass otolith. The color gradient goes from blue, which is low, to red, which is high. Starting at the top left, magnesium shows some promise, with some patterning present. Then continuing to the right with our control element calcium, which was homogeneous throughout the otolith as expected. Next is manganese, which also has some patterning present. Then back to the middle left, the next two, copper and zinc, show no pattern at all, which kind of removes them as potential candidates. This is then followed by some weak patterning in strontium to the middle right. Barium, however, on the bottom right, had the strongest signals, which seem to overlap with the translucent zone formation when compared to the plain otolith section on the bottom left. Taking a closer look at the barium 2D map, now on the top left, we can compare it to the line analysis that was taken along the white arrow via LAICPMS, the results of which you can see on the graph in the right. Each spike on the map coincides with a clear peak on the graph. This shows how barium would be a good candidate element for our Loms Gargle periodogram analysis. With barium revealed to be a good candidate element, we then proceeded to run our elemental data through the Loms Gargle analysis. On the left, you can see a graph of each barium line transects of the otolith standardized between each run on the LAICPMS. Then, each point in the analysis was assigned to its portion of the annuli. That data was then run through the Loms Gargle analysis, providing the periodogram on the right. This periodogram shows that there is significant periodicity present, but it is at the 1.6 year mark rather than our hypothesized 1 year mark. Repeat analyses are still being conducted to confirm whether that cycle for barium is actually at the 1.6 year mark, or if the analysis points from the LAICPMS were assigned to the wrong portion of the annulus. Currently, we are still trying to iron out some of the wrinkles of our process. In conclusion, 2D maps of structures did help reveal candidate elements. In black sea bass, barium fluctuated over the annular cycles at a rate of 1.6 cycles per annulus. Formation of opaque zones do occur cyclically, but that is inconsistent with the seasonal cycles we are seeing in barium. Revealing these cycles does help set the foundation for age validation in non-uniform species such as Atlantic monkfish. That brings me to our future work, where we are trying to apply this methodology to Atlantic monkfish, a species that provided that funky otolith on the right that prevents traditional aging and validation methods. Here are some preliminary results of this work using the Elysia, or angler, which is circled in red on our monkfish here, a section of which you can see on the left. 2D maps have shown that manganese to be a promising candidate element with distinct fluctuations in concentration, which you can see on the right. Lastly, I would like to acknowledge Drs. Dave Secor, Rose Jagus, Ann Richards, and all of my collaborators without which none of this work would have been possible. I would also like to thank NOAA, EPP, LMRCSC for funding this work and making this presentation possible. Thank you. Here are my references. Any questions can be sent to me at bfrey@umces.edu or on Twitter @sciencefry. Thank you for attending my talk.